everyone, and greetings from the Nanomedicine Research Group of ICT Mumbai. My name is Sanjana Krishnakumar, and I'm a science communicator working with the Nanomedicine Research Group. Joining me today is Dr. Sarah McMullen, who is the director of the Indian Office of the US FDA. So, Dr. McMullen, thank you so much for joining me today, and I'm so excited to have you over here. And how are you doing?
I'm doing great. Thank you for asking. And I'm really excited to be a part of the podcast today. So thank you.
I'm so happy to hear that. So, you are the director of the Indian Office of the US FDA. So, what does your role look like?
No, I'm glad you asked that. And, when I sit and think about what our office does, I think the better question is, what does our office not do? Because we cover such a wide portion of the economy, and it's actually really exciting. But it's also a bit of a daunting challenge as well. So, as our name implies, Food and Drug Administration, we do cover foods and drugs, but we do a lot more than that. We also do veterinary foods and veterinary drugs. We do biologics, medical devices, cosmetics, tobacco. If you think about it, FDA regulates it. We regulate 20 cents of every dollar that's spent by consumers in the US. So that gives us a lot to look at. And so on any given day, if you follow my LinkedIn profile, this month you saw me on a farm in Assam where we were giving training to spice farmers, or speaking on a panel at the IPA conference in Mumbai actually last month already. So it's a variety of things that we cover here. We do everything from inspections to policy to capacity building, and it's really exciting to be able to cover so many different areas, but also a challenge as well.
That's very interesting. And so you do travel to different parts of India as a part of your work, do you?
We do, and it's one of the best parts of this job. I've been to, I think, 18 Indian states so far, between my professional travels and my personal travels from my time here in India. And there's something special about every place that we've been able to travel. So that's been one of the things that's been really remarkable and helped us understand what's possible here in India and what some of the challenges are as well.
So I don't know if our audience also follows us on LinkedIn and Instagram and other platforms, but Dr. McMullen actually visited the Nanomedicine Research Group at ICT Mumbai last month in June, where she saw our various labs and facilities. And so does the US FDA like to visit a lot of institutes as well to, see what they're doing?
Yeah, so I think, that's one of the areas that we get to engage in here in country, is also understanding where technology is leading. In a lot of instances, technology evolves and regulations have to follow, right? So in order to keep as close a gap as possible between the evolution of technology and the regulatory oversight of that technology, we need to have an understanding of what's in development. And then as things become implementable, sort of how that happens, and sort of being able to apply regulatory concepts to innovations. And so visiting academic institutes such as ICT really gives us an understanding of what's possible and what's coming in the pipeline. So these are very helpful and useful visits for us as well.
So, as you said, a lot of research institutes, even ICT Mumbai, we are doing a lot of research and development and working on developing new technologies. So, in labs like us that do a lot of research like this, there are certain standards that are to be maintained, or good lab practices, as we teach undergrads and postgrads over here. So, could you tell us more about what those standards are that all these labs have to maintain while they're doing their research?
Yeah, no, that's a great question. And so really depending upon the end use of the data that's being generated, there's going to be different levels of stringency applied in terms of the regulatory oversight of that data. So when we talk about R&D data versus data that's going to be used in a submission, there's a little bit different expectations on the laboratory side, on the quality practices that must be in place. I'll speak some today because I know we'll probably get into discussions regarding it, but the integrity surrounding data generation, recording, and archiving is really important because if we can't trust the data, then that leads us to question sort of everything about what's being shown to us. So, while you have different levels of stringency in the expectations, the data quality should remain the same regardless of what the data is being used for.
That's very interesting. So earlier, when you were telling me about all the different activities of the FDA, you mentioned a term called biologics. So, as our audience has probably heard from our other podcasts, biologics are drugs that are developed using living cells or products of cells, correct? So, could you
Yes.
tell us more about what goes into the regulation behind the development of biologics?
Yeah, no, and biologics is quite a different entity than when we talk about traditional pharmaceutical manufacture, which is typically small molecule manufacture. So we're seeing a lot of manufacturers transition from small molecules towards the biologics. There's probably more biologics that are coming off of patent, and so then we see the biosimilar development taking place as well. These processes are very complex. It's not cookie cutter. Usually when you're talking about chemical reactions, you get sort of the same thing each time. You have a level of consistency that you expect. That's not always present in biological development and processes. Again, these are all sterile products as well, so there's a lot of additional considerations in the manufacturing process. So we have a lot of guidances for industry regarding biologic manufacture, but those are just some of the nuances that make it a little bit different than traditional pharma manufacture that require additional considerations and precautions.
All right. So, um, so have you interacted with any other labs that work with biologics or like manufacturing of biologic drugs?
We do. So we have, there's firms obviously that we inspect here in India that are manufacturing biologic products for the US market. And we also interact with a lot of incubators and different cells, C-Camp, Bangalore Bioinnovation Center. There's a lot of T-Hub. There's a lot of these places around India where there's a lot of technology and work that's going into the biologic manufacture and processing. So that's been, we've been able to engage with them and sort of understand where they're going. And then obviously we have the manufacturers that are already making these products as well.
So once these products have been developed by a certain lab or a certain industry, how do they make it to the market where they can reach a patient or someone who needs that particular drug?
So we have what's so like in drugs, we usually have what's called an NDA, new drug application, or ANDA, abbreviated new drug application. For biologics, we have the BLAs, the biologic license application. So that's a separate set of rules and processes, but they're similar, but they're specific to biologics. And so one of the uniquenesses about biologic manufacture is when we do a pre-approval inspection, it has to be when manufacturing's happening. So this requires a little bit of coordination when we do our inspections of these facilities prior to them getting approval for a product on the market. We actually have to see them make the product. So for like small molecule manufacturers, they can do validation batches ahead of our inspection. And so then we retrospectively look at the data. But for biologics, we have to see it in action because it's such a complex process. And there's so many nuances associated with biologic manufacture that aren't necessarily there for small molecule. So, and then with a successful inspection and application process, then that product can make it to the US market.
Nice. So do these do biologics and other small molecules also have to go through clinical trials before they make it to the market?
They do. So depending upon the product, if it's a new product or new biologic, obviously the data generated for a new product is going to be different than something that's being produced or presented as a biosimilar. So we would look to, there's different clinical trial requirements, which we have guidances and regulations that clearly explain the differences. But we would expect to see them perform in a clinically similar mechanism to the innovator product. And so that's what a lot of these biosimilar and also on the small molecule side, the the bioequivalence or bioavailability clinical trials, are really to demonstrate that the product has the same efficacy as the innovation product.
So the biosimilars are generally made in reference to a reference drug or your biologic, I mean reference biologic. So if so is there a chance that is there a chance that your reference biologic would have been tested a, on an animal model, and when you're developing a biosimilar that's going to be tested on something entirely different, and in that case how would you ensure that they both work in a similar way?
So, and I know we're talking some about animal studies, but in general, the level of evidence would not be that different between an innovator product and a biosimilar product. And so whoever is manufacturing the biosimilar, if they wanted to present different levels of evidence compared to what would have been presented for the innovator product, they can have that dialogue with the agency. We have guidances in place, and then we also recognize that there's unique scenarios and situations, especially in the world of biologics. And so in the actual BLA process for biosimilars, they have opportunities to have meetings with the agency at routine intervals because there are things that come up and there's questions that manufacturers want to understand and be able to resolve before investing money in clinical trials or further development. So recognizing the complexities in this area, FDA has these opportunities to engage in the earlier in the process.
Right. So as we're talking about testing and developing drugs, there's also a lot of talk about incorporating methods of like doing preclinical testing on models other than animal models. So things like organ on a chip or spheroids. So have you seen any instances of that being in the part of mainstream drug development, either in India or in the US?
Yes, I think, when we talk about alternatives, I think there's a few areas I would like to highlight. So one of them is in silico modeling, one is cell-based assays, and the other is in vitro microphysical systems. So when we talk about things like in silico modeling, this would be using information in computational science approaches to predict safety issues or to model performance in a living system. So this could be done to supplement and maybe potentially replace risk analyses that are currently based on animal data. So we're still looking at the predictive value of current non-clinical testing approaches, but it's not always possible to use modeling to remove the need for animal models. I will point to one area that we have active research ongoing in, and it's called, we established it in 2021, and it's called the Open Online Simulation for Stimulating Peripheral Activity to Relieve Conditions. That's a really long title, but this was actually a platform that FDA developed and made available that enables neuroprosthetic developers to simulate modulation of the peripheral nervous system and its impact on organ physiology. And so this is using validated models and state-of-the-art in silico tools. So this is actually something that's available as people are developing things so they can test it out in this in silico way. Another alternative, as I mentioned, was this cell-based assays. And so these can be used for things such as neurotoxicity. And this is some of the broader work that we do to evaluate more reliable systems and biomarkers for monitoring toxicities, side effects, and abnormalities. So for this particular scenario, developmental neurotoxicity tests are needed for sensitive populations, such as infants and children. And neurotoxicity can occur when the nervous system is purposely or accidentally exposed to toxic substances. And so this could be chemotherapy, this could be lead, mercury, other things. And so this is one of the alternative methods that FDA is researching. And we use in vitro cultures of developing brain stem cells, since there's no currently established predictive in vitro neurotoxicity assays that allow correlation to neurodevelopment in pediatrics. So this is just one of the examples for the cell-based assays. And then lastly, I'll touch on the one that we saw at ICT Mumbai. These are the in vitro microphysiological systems, or these organ on a chip. And this is another modality that's shown potential to produce predictive data. to support development of human and veterinary products. And so we see many models being worked on, including cardiac, renal, hepatic, and as we saw at ITC Mumbai, ocular, pulmonary, and dermal. And so on this front, we understand that there's several robust centers of academic research in creating and testing these models. And so we're looking to develop performance criteria in order to provide realistic assessments of the potential of these new tools for safety and efficacy testing. So these are some of the ways that FDA is looking at alternatives. And we really focus on what we call the 3 R's, and that's to replace, reduce, and refine our dependence on animal studies. So those are just a few of the examples of the work that's going on with the agency.
That's very, very interesting. And thank you for explaining that so well. Do you think that there can be a day when we actually stop relying on animal models entirely, just with in silico models and organs on chips and spheroids and other forms of in vitro systems?
I think that's the hope. We're not there yet, but I think that's definitely the hope. And what we've seen with technology and the way that technology evolves, even what we're able to do with machine learning and AI, really understanding and predicting information is becoming much more attainable than it was previously.
But what do you think are some of the challenges with using animal models that's kind of pushing researchers to using in vitro models or in silico models when it comes to testing drugs or biologics?
I mean, like others, the humane treatment of animals is increasingly a concern in the research community and in FDA. So, what we've historically used animal studies for are for the risk, safety, efficacy, and public health impact of regulated products. And for that, we've really focused on the product's nature, the chemistry, the pharmacology, and the toxicology. So for, kind of putting it another way, we use animal models because they are systems of increasing complexity that help us understand how novel drugs or therapeutics behave in humans. And so this has helped us be able to manage the risk to humans and to patients. That being said, it does come at a cost, like you mentioned. These are the ethical concerns with animal experimentation, and then also the resources needed to ensure animal welfare. We make sure that animal welfare needs are adequately addressed. For instance, in the US system, we try to ensure the welfare of primates that are used in animal testing well after experiments are ended, often for the duration of their lives. So really, again, our focus here is to, like I said, our 3 R's, to reduce, replace, and refine the dependence on animal data. And so, obviously the humane treatment of animals and also the ethical concerns are things that are really driving that work forward.
That's very interesting. And I really do hope that, we all move towards research that is not heavily reliant on animal models. That would be really great. So I remember when you came to visit us at ICT Mumbai, you told us about all the activities of the FDA, and you also mentioned certain opportunities about people collaborating with the FDA or joining or learning with the FDA. So could you tell our audience more about those?
So in India, we do have some engagements or some opportunities in India. More of our positions and our opportunities to work for FDA as an employee are in the US, with good reason because we're a US-based institution. But that being said, there's a lot of opportunities to work with the agency across the sectors. As we mentioned earlier, for us, it's important to understand how technology and innovations are evolving. So for that, we work with academia. We also work with manufacturers. A lot of manufacturers have R&D cells or are working with incubation cells. That helps us understand where the work is going. And we also, there's a lot of opportunities between our governments. There's Fulbright scholarships. There's a lot of different opportunities for studying abroad and different programs like that. So while not specific to FDA, there are a lot of opportunities between our two countries to learn and to do work that really is impactful for public health.
Anyway, thank you so much for speaking with me today, Dr. McMullen. I learned a lot from this conversation, and I'm sure that our audience who would be listening to this is also going to learn a lot. And I hope you have a great day. And once again, happy Independence Day to you, because we're recording this one day later.
Yes, thank you so much. And, you know, we're always happy to have people contact us. I'll give you my email address. It's for our office here. It's sorry, let me start this over again. US-FDA-INO at fda.hhs.gov. So if anybody has any queries, FDA related queries, we're happy to have them reach out to us, and we'll get back with them. So thank you again.
Thank you.